Okay, here we go. So please write your questions in the chat box and we'll save some time at the end to try to answer them. Many of you may know Dr. Seth Pollack from his work in immunotherapy in Seattle. This fall, he became the director of the Sarcoma program at Laurie Cancer Center in Chicago at Northwestern University, known on Twitter as ImmuoSarc. He has won awards and has been a lead investigator in clinical trials for immunotherapy and sarcoma. We're grateful that he's here with us today. So welcome, Dr. Pollack.
Hi, thank you so much. I'm really so grateful for the invitation to be here. Let me, can you see my slides?
Yes.
Okay, great. Yeah, no, it's really great to be here. So, you know, I'm going to be talking to you guys today about some of the basics in immunotherapy. And you know, I know you guys have a really educated group, and I know that you guys have had a lot of really star researchers talking with you guys,
Okay.
and Brian and all these people. But you know, so I'm going to try and balance it out because I know that you guys are an educated group, but I'm also going to try and make this a presentation that pretty much anybody can follow along with. So, you know, we're really going to be talking about the basics for immunotherapy for leiomyosarcoma, but in some ways it'll be, you know, very cutting edge because we're going to talk about kind of how a lot of these things that are really fundamentals of immunotherapy apply to leiomyosarcoma specifically. Okay, so yeah, I think everybody is excited about immunotherapy, right? You know, it's on the cover of Time. It's something that has really changed oncology, and even just, you know, watching the ads on the television, you see advertisements for really exciting immunotherapy, and I think it's been great. It's been great for oncology, but it's also been frustrating for those of us who are trying to make these treatments work in sarcoma and leiomyosarcoma specifically, because things haven't moved as fast as we would like them to move. So really the fundamentals of all immunotherapy come down to the T cells. T cells are really how, where the rubber meets the road when we're talking about immunotherapy, no matter what sort of immunotherapy treatment we're talking about, whether we're talking about checkpoint inhibitors, cellular therapies, vaccines, and I'm going to talk about all those different things today. But really the thing that kills the tumor at the very bottom is the T cell. And the way a T cell recognizes a tumor is with a T cell receptor. So every T cell has, I'm going to simplify it a little bit. Pretty much every T cell in your body has a different T cell receptor. Of course, it's not every, you know, there are groups of T cells, but, you know, the number of different T cells that you have is astronomical. It's like on the order of 10 with 12 zeros after it. And each one is programmed to see a different thing. So it's not that the T cell can just naturally tell what is foreign and what is supposed to be there and what's not supposed to be there. All the T cells that should see what's supposed to be there have been gotten rid of, ideally, and all you should have left are T cells that can recognize viruses and that can recognize tumor. And every T cell is sitting there, and for its whole life, it may go and not see the thing it's supposed to see. But sometimes it will see the thing it's supposed to see. And that could be a viral protein. That could be a mutated protein. That could be a normal self protein that's just not supposed to be there, but in the cancer starts becoming expressed. So the T cell is there with its T cell receptor in yellow here, and the protein is here, it's in red, and that's been made inside the tumor cell, and it gets presented on the cell surface by what we call the MHC complex. And the T cell just goes from cell to cell every day, your whole life, looking at different MHC peptide complexes. And when it finds the one that it's looking for, it kills the target, it kills the tumor, it starts to divide, it releases factors into the area that are called cytokines that recruit other T cells to come into the tumor, and also change the other cells that are in the area. Hopefully it makes other tumor cells that are adjacent also more receptive to T cells. And help them show their red circles better to the T cells that are circulating around. But their T cells are an important cell in, they're the critical cell in cancer immunology. You know, I'll say just a word about B cells. B cells are, those are the cells that make antibodies. And sometimes people will ask me, do you consider an antibody immunotherapy? And it's a little bit complicated because, of course, antibodies are part of the immune system, right? In the old days, I think a lot of people would call the antibodies as being immunotherapy. But these days, I don't personally consider, or when I'm talking about immunotherapy, I'm not talking about drugs like Herceptin or rituximab. These are antibodies that link directly to the cancer and kill the cancer directly. And really what I'm talking about are therapies that are aimed at manipulating the immune system somehow. So for example, drugs that are targeted to PD-1, they are antibodies, but you know, really the way you use them is to activate the T cells. So that's kind of the distinction that I make when I'm talking about immunotherapy. And that's different than, you know, B cells are probably important for immunotherapy because you know, there have been some studies that have seen increased B cells in sarcoma patients that respond to immunotherapy. And I think that's a really important finding that everybody's really interested in. Natural killer cells are another type of immune cell, and they're very closely related to T cells. T cells, as I showed you before in the prior slide, T cells can see their target antigens, the red circle here, if there's an MHC complex here. But what if there's no MHC complex? What cell is looking for a cell without an MHC complex? Because that's actually a common strategy of both cancers and virally infected cells. They get rid of all their MHC complexes so the T cells won't see them. And when that happens, the natural killer cells, it's their job to find the cancer. So they do play a really important role in cancer immunotherapy. I would say that we're just starting to really get a sense about what the potential for natural killer cells is. That's something that we've just scratched the surface of. T regulatory cells, they're a type of T cell, and they see their antigen the same way that T cells do presented by the MHC complex, except they do the opposite thing when they see their antigen. They tell all the other T cells in the area to go away. And for some tumors, we think that's an important way that they have immune escape, is the T regulatory cells that are in the tumor tell T cells to go away. In leiomyosarcoma, I don't think that happens. In leiomyosarcoma, we've looked for regulatory T cells or Tregs, and we haven't seen that many. So the levels of Tregs in leiomyosarcoma is very low. That doesn't mean that there's no role regarding Tregs, but I don't think they play an important role. But what I think we have seen, and you guys know really well, because you guys have worked closely with Matt Van der Rijn and that group, are that these myeloid cells, and particularly the macrophages, play such an important role in leiomyosarcoma. Leiomyosarcoma, we know these tumors have lots of macrophages. And if your tumor has macrophages, and those macrophages have some of the what we call the bad markers, or what somebody might call bad in quotes, that portends a worse prognosis. You know, there's different types of macrophages. So we have macrophages play a really important role in the body. So if you have, they do all sorts of things. So one of the things that they do is they clean up debris. So if you have like a burn or there's a wound and there's all sorts of like healing tissues, dying tissues, the macrophages come in, and one of their jobs is to clean up all of the junk. And what they do is they sort through the junk and they look for something that could be important to the immune system. And if they see something that's important to the immune system, they can show it on their surface of the macrophage to the T cells. And then the T cells will say, hey, look, what is this? And then they'll go and look for whatever that is. And for example, if it's a tumor, maybe they'll try and kill that tumor. So that's kind of like one way the macrophages could be. That's like the ideal way. That's a M1 macrophage. So M1 macrophage is looking for dead cells, dying tissues, and if they find something concerning, they will alert the T cells. They can also produce cytokines that recruit T cells into the tumor and kill the tumor. But the problem is there's another way the macrophages can be. You can have what's called this M2 macrophages. M2 macrophages are really important for wound healing. In some situations, you might have a wound, and any wound is going to get some foreign material in it. And it might be that at this moment, you don't want a big immune response in that wound because you're trying to heal, right? You don't want like all this infiltrate in there, right? You want some, just calm down right now. We don't need a big inflammation. We're trying to heal right now. And these M2 macrophages are what we call more wound healing macrophages. They coordinate the healing response, and they tell the T cells to go away. We're busy healing. We don't want inflammation right now. And the problem that we see and that we think is really important in leiomyosarcoma is that the macrophages in the tumor are polarized to this M2 phenotype. So they think that they're in a wound and they are telling the T cells to go away. We don't need to have an inflammation right now. And that's what's going on. And this is a simple cartoon. So the truth is that all these macrophages kind of exist on a spectrum. And that's why I like this picture, because it's not just that they have to be orange or green. They can sort of be like a mix. But in leiomyosarcoma, we think most of the macrophages are winding up more on this side of the diagram. What about PD-1? So PD-1 is kind of the biggest immunotherapy that you guys are all hearing about. This is, if you guys, are watching sports, like I like to watch sports, they always seem to have commercials for Keytruda and for Optivo. Those are two of the biggest blockbuster treatments, PD-1 treatments that are FDA approved for so many different diseases, melanoma and lung cancer and the like, and they probably work really well. We think they work really well for certain sarcoma subtypes like undifferentiated pleomorphic sarcoma or alveolar soft part sarcoma. Those are some sarcomas where the role of PD-1 is undeniable. But in leiomyosarcoma, the role of PD-1 inhibitors is controversial, and we really haven't figured it out yet. And I think we will figure it out. So the way PD-1 inhibitors work is that you have a T cell, and the T cell is seeing the tumor cell. And in this case, the tumor cell has presented the antigen that the T cell recognizes on its cell surface, but the T cell has an off switch, and that is PD-1. And this tumor happens to be expressing PD-L1 on its cell surface. And when the PD-L1 interacts with the PD-1, that turns the T cell off. So if you have a drug that blocks PD-L1 or blocks PD-1, that will prevent the T cell from getting turned off and allow the T cell to kill the tumor. It's not an end all and be all, because there's lots of ways that the tumors avoid T cells killing them. This is just one of those ways. And for some patients, it's really important. For sure, in some of these very inflammatory tumors like melanoma, lung cancer, this is it. This is like the whole thing that is keeping the T cells from wiping out the tumor. And if you could just block that, you're done, you're home free. That's not the way it is in leiomyosarcoma. It's almost never that way. This is almost never in leiomyosarcoma the one thing that is preventing immune destruction of the tumor. But I think that there probably are some patients where this is one of a few things that is going on. And for those patients, it may make sense to have some sort of immunotherapy regimen that involves a PD-1 inhibitor. Now, a lot of you guys know about the SARC-28 study. This was kind of one of the classic definitive looks at, or it was an initial look, but it sort of set the stage for how we think about immunotherapy in soft tissue sarcoma. And they looked at 10 leiomyosarcoma patients, 10 undifferentiated pleomorphic sarcoma patients, 10 liposarcoma patients, 10 synovial sarcoma patients, and they looked to see how each of them did with just a PD-1 inhibitor. And what they found is that with undifferentiated pleomorphic sarcoma, you could see a lot of these patients responded. And when they responded, these responses were pretty durable. Dedifferentiated liposarcoma didn't quite have so many responses, but there were quite a few patients that seemed to do well for a little while. At least they had their disease controlled. The leiomyosarcoma patients are in blue, and you can see these patients did not do as well in the SARC-28 study. They tended to progress, and it was really frustrating. There's also this trial that was done at Dana-Farber looking at uterine leiomyosarcoma, and these patients also all progressed. There were no responses here. But I mean, there have been patients with leiomyosarcoma that responded. So this is another patient also treated at Dana-Farber who had a great response to a PD-1 inhibitor. This patient also progressed, and it was very interesting because they found that there was a mutation after her progression. But this, you know, I really take a lot of heart from this because there are leiomyosarcoma patients that progress, that respond to these treatments every now and then. So, you know, it is something that we think about sometimes. Sometimes people ask me, do leiomyosarcoma patients ever have PD-L1 expression in their tumor? And they do. So there are quite a few patients with leiomyosarcoma that have really strong PD-L1 expression. There are also some that don't, but there are some that do. And some of them have intermediate levels of expression as well. We also, you know, we see a similar pattern of expression when it comes to T cells that express PD-1 inside the tumor. So, you know, not that the PD-L1 expression is the be-all and end-all of whether you would respond to PD-1 targeted treatments. For sure, patients with very strong PD-L1 expression don't always respond. And the converse, you know, in melanoma and lung cancer, they see patients that don't have any PD-L1 expression and PD-1 expression in their T cells. And I don't quite understand it, but sometimes those patients will respond to PD-1 inhibitors, and it's probably just, you know, they just need something to get them over a hump. You know, just a little bit of immune reaction sometimes is the spark that you need to light the fire, and then all the T cells come in and start to kill the tumor. So it happens for sure. Yeah. Oh, this is the SARC-28 study again. I'm sorry. Oh, so, you know, I put this slide on because I think a lot of you guys have seen this. This is a Kaplan-Meier curve, right? So this is the percentage survival, and this is the years, and you can see this is the if you have a very bad macrophages, you know, the survival kind of drops really quick. Whereas if you have good macrophages, you have a longer survival. And there's a number of studies right now that are trying to combine PD-1 inhibitors with drugs that can deplete macrophages in the tumor. I mean, that was part of what we were thinking about when we put together our doxorubicin pembrolizumab study that was published at the end of last year. We had a trabectedin avelumab study. So trabectedin, a lot of you guys have probably been on trabectedin. Trabectedin is an FDA approved drug for leiomyosarcoma and liposarcoma. It's chemotherapy, but it also has this other way that it works, which is by killing the macrophages in the tumor. And the trabectedin is just really, really potent at killing macrophages. And that's been shown really, really well. And because of that, we launched a study that, and others have launched similar studies based on our ideas using combining trabectedin with a checkpoint inhibitor. And so we had this study combining trabectedin with avelumab, which is a drug that blocks PD-L1. We had some really good data, and I think that we're going to be able to tell you guys all the details about the results of that study later on this year. But some people, like Sanchala from Santa Monica, he's presented some data with just a small set of patients. I don't think that there's a really full data set of patients that's been presented. So I don't think that we can really say how effective that combination is. I mean, I can tell you just anecdotally, we did have some really durable disease regression on our trial, which is really nice. I mean, that's kind of what you want on a trial like this. But I don't think that anybody has really seen a data set looking at where you can really kind of say some statistics, especially not in leiomyosarcoma. Sanchala's short data sets that he's presented have been, 8 patients and, maybe 1 patient's had leiomyosarcoma or something like that. And they've been in very refractory settings. So I think the jury's still out, but now, and you guys know this too, that there's a whole slew of combination of chemotherapy and checkpoint inhibitor trials that are going on right now. And I know you guys supported the really fantastic, excellent work of Dr. Mova from Memorial Sloan Kettering, and she's interested in combining PARP inhibitors with checkpoint inhibitors. So PARP inhibitors are not, I mean, it's a different type of chemotherapy. They're really not, that's kind of a different concept. Rather than going after the macrophage, it's more like trying to create more mutations in the tumor. But I think there probably is a secondary effect of affecting the macrophages whenever you use some sort of chemotherapy type of drug. So I think all of these checkpoint inhibitor combination trials are going to be coming out in the next couple of years. And I mean, I think there's going to be really good data coming out in the next year or so. And I do think that combination chemo immunotherapy is going to be one of the next waves for leiomyosarcoma. I think it's going to really make a difference for a lot of leiomyosarcoma patients. Let's see. Yeah. So, this is just from our doxorubicin and pembrolizumab trial. We did have some patients with leiomyosarcoma who had continued response after we pulled off the doxorubicin. Our patients did pretty well with doxorubicin and pembrolizumab with leiomyosarcoma. We looked and we compared the data from our historical data using just doxorubicin alone, and there was actually it's hard to do this sort of comparison, but even when we just focused on the leiomyosarcoma patients, the patients on our trial did a little bit better. But I don't know for leiomyosarcoma whether it was enough to make a difference, but to make the kind of difference that we would need to see to really sort of switch all of our leiomyosarcoma patients over. But I think it was intriguing enough that it really supports some of the other ongoing studies. For example, at University of Colorado right now, there's a study that combines ipilimumab and nivolumab, a CTLA-4 inhibitor and a PD-1 inhibitor, along with doxorubicin. So I think that these things are moving, one foot in front of the other, and we're making progress. So a lot of you guys have heard about Jim Allison. He won the Nobel Prize, I think it was a couple years ago now. He's at MD Anderson. He did a lot of his groundbreaking work before that, he was at Berkeley and he was at Memorial Sloan Kettering, and he's the one that discovered the first immune checkpoint. So the first immune checkpoint wasn't PD-1, it was CTLA-4, and that is the target of ipilimumab, or Yervoy is the drug that you guys probably know about. Ipilimumab is sort of the scientific name, Yervoy the technical name. CTLA-4 is I don't want to get too deep into the weeds, but it's a break. It works a little bit differently than PD-1, but it's kind of the same basic concept. So we have a antibody that blocks CTLA-4, and it allows the T cell to be activated. And there was this great study from the Alliance that looked at the combination of anti-PD-1 and CTLA-4, and they did include a bunch of leiomyosarcoma patients, and they seem to find better. I don't know if they found better results. I mean, I thought that I personally think the results using the combination of Ipi-Nivo with nivolumab are, I think it looks a little bit better to me. This is a waterfall plot. So the way you read a waterfall plot is this is the best response for each patient. So each line represents a patient, and this is the y-axis is the biggest, the best change in tumor size. So like if you're over here where the line goes all the way up to 100%, that means that this patient progressed right away. Whereas if you're on the right side of the waterfall plot and your line is going down, that means your best response was that eventually your tumor shrank by more than 50%. You can see that the Ipi-Nivo combination seemed to have more really fantastic responses. And you can see that on the Ipi-Nivo combination, it seems to me that the patients seem to be able to stay on their treatment a little bit longer. So, we're still sorting out everything that this means, but to me, I think that it's encouraging. I think it means that although the data originally with just single agent PD-1 was a bit disappointing, there's a lot of room for improvement, and we're doing it, right? We're having patients on these checkpoint inhibitors with leiomyosarcoma for a while, in some cases, when we try these different treatments. Oh, this is a Kaplan-Meier curve. So that means that the x-axis is the time, and the y-axis is the percentage survival, and the red is the overall survival. And you can kind of compare the chart on the left and the chart on the right, and it looks like the percentage survival line in general is just a little bit higher on the chart on the right compared to the one on the left. This study was not done in a way where you can really compare the two, but we're all kind of comparing the two a little bit. What about T-cell therapies? A lot of you guys have heard about cellular therapies where we genetically engineer T-cells to fight cancer. And it's happening right now in certain sarcoma subtypes. It's happening in synovial sarcoma. It's happening in myxoid round cell liposarcoma. It's happening in Ewing sarcoma and osteosarcoma in some clinical trials at some specific locations. It's not happening yet in leiomyosarcoma, but I think it will. There are two types of cellular therapies. The first is called a CAR, and the CAR T-cell therapies are sort of the biggest blockbuster T-cell therapies, and you probably heard about them. They have totally transformed the treatment of patients with certain types of leukemias. So certain types of leukemias, especially their pediatric leukemias that were just terrible, guaranteed death sentence just a decade ago. Now these patients are being cured in a majority of the cases because of T-cell therapy. It's amazing. But they haven't been as that effective in solid tumors. There are some lymphomas where they seem to work some on. There are some other types of leukemia that they seem to work on, but in solid tumors, they haven't worked that well. The reason why these things are called a CAR is because the CAR stands for chimeric antigen receptor. So chimeric means a combination of two different types of things, like the chimera in mythology combines lions and dragons and things like that, right? In science, right, the chimeric antigen receptor combines two things. It combines the inside of a T-cell receptor, the T-cell receptor that I showed you before, that looks for the antigen and the MHC complex. But instead of having on the outside, the T-cell receptor has an antibody, and it's looking for something abnormal on the tumor cell surface. The good thing about that is that if you find the right thing, it's really, really effective. The bad thing about that is it's really hard to find new things on the tumor cell surface that you haven't found before. So there are CARs that are looking at things that are expressed by leiomyosarcoma. For example, we have a lot of really good data showing that B7H3, that's a protein that is a very cancer-specific protein, that's been seen in a lot of different types of cancer is pretty much always expressed by leiomyosarcoma. And there is a B7H3 CAR. It's being tested at Stanford. It's not being tested in leiomyosarcoma. It's being tested in osteosarcoma and Ewing sarcoma, but I think it will get there. I think it will get there. And I think that there are a number of similar types of CARs that will eventually get there. TCR is a little different. It's one of my big pet peeves that a lot of the times I hear sarcoma people and even people who should know better calling TCR therapies CAR therapies, because TCR is really a TCR, like I showed you at the beginning. It's actually the TCR from a T cell, except it's like a TCR from a specific T cell. And in this T cell, it works really good at killing cancer. And what they do is they take that TCR, maybe they modify it just a little bit to make it work better, and they put it in other T cells. And that's what's happening in synovial sarcoma and mixed red round cell liposarcoma. I don't call those CAR therapies, even though a lot of people do. It's not actually technically accurate. Those are TCR therapies. Regardless, it would be great if we had one for leiomyosarcoma. Leiomyosarcoma just doesn't frequently express the targets that we see in synovial sarcoma and mixed red round cell liposarcoma. So usually in those diseases, we're targeting proteins from the specific family of proteins called cancer testis antigens. And those include some proteins that you may have heard of, like NY-ESO-1, MAGE, A4. Those proteins are seen a lot in those sarcoma types. They're not seen in leiomyosarcoma. I pretty much say they're just not seen. Now, I've treated a lot of patients with these sorts of therapies. I actually have treated a few leiomyosarcoma patients, but even the leiomyosarcoma patients that I did treat with treatments targeting NY-ESO-1 and MAGE-A4, you know, in synovial sarcoma and mixed red round cell liposarcoma, those proteins are expressed really strongly throughout the entire tumor. In leiomyosarcoma, it's just not like that. Even when those patients, those rare patients, have some expression of it, it's like a little ditzel in a corner of the tumor. It's very patchy. It's not strongly expressed throughout the tumor. So I just don't think those targets are good for leiomyosarcoma. If you hear about NY-ESO-1 targeted trials, MAGE-A4 targeted trials, it's great for those sarcoma subtypes, but unfortunately, it's just not, they're just not for leiomyosarcoma. That's just how it is. This is just showing, you know, so the most common target with these CARs is called CD19. This is showing how miraculous they can be in lymphomas, PET scan. You know, some of you guys have had PET scans, and you can see, you know, the yellow means that they're taking up a lot of radioactive glucose, and you can see that after treatment, that's all gone. Before treatment, these tumors have lots of, this is a liver, right? And that's, the red is a normal background color for liver, because livers in your normal state, they're always using, it takes a lot of sugar to run a normal liver, but not quite this much, right? This yellow spot is really, really intense. So that's like, this is a really, really hot tumor, and it goes away completely after treatment. And here as well. So this is just to show you how exciting these CAR T cells can be. And I think there will be a time in the future when we start using them for leiomyosarcoma. We're not there yet, but we're on our way. So some other treatments that people talk about is part of the new wave of immunotherapy. There's some really old drugs that have been around forever. I told you before about cytokines. Those are the signals that the T cells use to communicate to one another, right? Cytokines, for example, if a T cell gets into a tumor, it'll release things like interferons and IL-2, and these signals attract other T cells to the tumor. And one of the first immunotherapies was IL-2. People would give high-dose IL-2 to melanoma and renal cell carcinoma patients, and sometimes those patients would be cured. It was unusual. It was like less than 10%, but it happened. So we would try it all the time, just because we'd be looking for that like one hit, you know, maybe this is the patient where we see that. But it made people enormously sick. People would go to the hospital to receive these. You had to be watched in the ICU just to receive the treatment because it made you so sick. nowadays, these things have kind of moved into the late, I wouldn't say they're not used at all, but it's very rare that they're being used even in melanoma and renal cell carcinoma because we have better treatments. But there's a lot of the really smart biotech companies are trying to come up with ways maybe we can make these things a bit more tolerated and more effective by making small alterations to them. And I think there's going to be a wave of trials. I mean, there already have been some trials, but I think it's going to be a bigger wave where we see these sorts of drugs combined with the PD-1 inhibitors that you've seen and some of the other immunotherapies that I think everybody gets really excited about. Cancer vaccines. So I think cancer vaccines are kind of one of the few things that are going to come out really stronger after COVID, because COVID has put so much, has pumped so much life into the vaccine research community. A bunch of vaccine technologies that people have been wanting to test for many years. They just haven't been able to get into patients. So for example, I've got a good friend who works at it's a cancer vaccine company. That's why it was founded. It's like everybody in there, that company really joined to do cancer vaccines. And they have, you know, a really novel gene editing platform, but it's like very cutting edge. So, you know, they had to go through a whole lot of stuff with the FDA to get into a clinical trial with humans. But when COVID happened, the FDA kind of said, look, you know, why don't you just go for your trial with a COVID? You know, we've really got to figure out COVID. And so just go for it, do your safety testing. So now, you know, of course, they're not like going to be one of the Moderna and Pfizer and these sorts of things. Their vaccine is not getting FDA approved, but they've had the chance to test the safety and shown that the basic platform is safe. And now they're going to be able to come back with, you know, all the other things that they want to try in order to vaccinate people for cancer. I've been involved in a lot of cancer vaccine trials. They are, I mean, it's frustrating. Cancer vaccines, vaccines just work really slowly. And I think that they are effective at inducing an immune response. But I think the thing, you know, usually when you get a vaccine against a virus, you know, your body doesn't have the virus in it. And it's really just preparing you for the virus to come. And cancer vaccines, it's sort of tough to vaccinate people against, like, you know, who have minimal disease, to vaccinate them against the cancer from coming back, because it's hard. There's a lot of kind of logistical issues with the clinical trials to sort of prove that you've had that effect. But I think we're getting better at the cancer vaccines. I've had a patient who had almost a complete response to a vaccine against NY-ESO-1. And I think that there's going to be, especially combining the vaccines with some of these other immunotherapies, could really just, you know, the idea is, what you really want is to launch a big immune response against whatever the antigen that's in the vaccine. A vaccine, I mean, really what a vaccine is it's a tool to direct your immune response against a certain antigen. And people are getting really creative at creating personalized vaccines. So, you know, you get, just like right now, I bet a lot of you guys have had Foundation 1 tests or Tempus or Caris, and you got like kind of a personalized report about sort of what genes are. And in leiomyosarcoma, unfortunately, most patients, sometimes patients have something that pops up on those reports, but for most patients, unfortunately, there isn't a lot of useful stuff that pops up on those reports. I mean, we do them because sometimes we find something, but, you know, unfortunately, the majority of leiomyosarcoma patients don't have something like that we could target. But what I think will happen is you'll get a report like that, you know, this is 10 years from now, and it'll basically be like kind of like a personalized sort of like vaccine type of report that they can make a personalized cancer vaccine to, and you'll be able to do that in combination with some other kind of immunotherapy, and they'll make a vaccine specifically for you, because they'll see, okay, you have this mutated protein, you have this mutated protein. These are both things that we can easily put into the vaccine, and just like they're targeting the spike protein for COVID, you know, they'll just put it in and, you know, make a vaccine for you, and you can combine that with the other immunotherapy immunotherapies that you're getting. So that's the vision of the future for cancer vaccines. There are cancer vaccines that are proven in certain cancer settings. There was one proven in lymphoma to work really well, although that kind of fell out of favor because there were just some better treatments in lymphoma. This is an FDA-approved, Provenge is an FDA-approved treatment in prostate cancer. It's for patients with metastatic prostate cancer who don't have disease that you can see on imaging studies, but they have, in prostate cancer, there's a good biomarker in the blood called PSA, and they have a rising PSA. These are patients who we think maybe, we were hoping maybe they were cured, but then, we see the rising PSA. But we don't see the lesions, and that's a good time to vaccinate these patients. And we know that if you give them Provenge in the right patient population, they seem to do better. And there's different types of vaccines, right? There's just some vaccines you're giving a virus, and the virus has been engineered to have a cancer protein in it. Sometimes we'll just give a peptide. So the peptide is just like a protein that we know is in the virus, but some vaccines could be like a cell, and the cell has the protein that we're interested in it, and it's presenting the antigen to the T cell. So there's a cell, and it's loaded up with cancer-specific proteins, and it's going to show those proteins to the rest of your immune system. And that's what Provenge is, with a prostate cancer-specific antigen. Toll-like receptors. I think a lot of you guys may have heard about William Coley. He was this orthopedic surgeon at the turn of the century, turn of the 19th to 20th century. He was injecting streptococcus into people's into people's tumors. And it was very controversial because he would actually, there were some people that died because of the sepsis that the streptococcus induced. But there were patients who had tumors that regressed because of the immune response that the bacteria induced. And this is called your innate immune system. So, the T cells, the antibodies, these are things that we call the adaptive immune response. The adaptive immune response means that whatever threat nature throws at us, whether it's COVID, whether it's the next virus that pops out of the woodworks, there's always going to be new things that are coming at us. Ebola, our bodies are just incredibly adept at reacting to something new and making a new immune response against it. But there are other things in nature that have always been there. They've always been threats. Certain bacteria like Streptococcus has always been there throughout biological history. Certain fungi have always been there. And your body just has like built-in traps that are specifically designed to kill those things. And when those traps get set off, it's like your body is pulling a fire alarm, and you start to release cytokines in the area where those things happen. Your immune system rushes in. They say, oh my God, there's bacteria here. We need to get the T cells into that area. And that's what happens, right? So that's what William Coley did by injecting bacteria into these tumors. It was like pulling a fire alarm for the immune system. Hey, look, something's going on right here. Guys got to check it out. And so that's what happened. So people are really interested in exploiting this effect. And maybe it doesn't have to be like William Coley, but maybe we can do it in a really safe way that also induces the kind of same type of response. There's actually a really good company that I've been partnering with that we're trying to take something forward with that it makes a very specialized killed bacterial cell product that is done really safely, and it's gonna trigger all of the toll-like receptors. I had a trial with another company that I used to work with, and they had sort of isolated part of the bacterial cell wall that triggers one of these. They're called toll-like receptors, is one of the receptors that's on all the cells in your body that is specifically designed to detect certain types of bacteria. TVEC is a genetically engineered virus that you guys may have heard of, and it works in a couple ways. So it's engineered. So this is an FDA-approved treatment, not for sarcoma, but it's FDA-approved for melanoma. And there's exciting data in sarcoma where people have seen some responses in patients injected with TVEC who also received a PD-1 inhibitor. So you have to actually have a tumor that somebody can inject directly with this virus. And the virus is engineered to produce GM-CSF, which is the same thing that you guys get if you guys get chemo, you know, that shot you come back, the Neulasta, the Neupogen, that's GM-CSF. So, but it's coming, but the virus expresses it. So the virus is producing it in the tumor so that the immune cells are not just being attracted everywhere like the Neulasta makes them go. They get pumped up in the tumor. So white blood cells are attracted to the tumor, and the virus itself is also immunogenic. It infects the cells. T cells want to kill anything that's infected with the virus, so they try and kill the virus. Is called an oncolytic virus. A virus, oncolytic means onco, like cancer, lytic, kill, right? So it's an oncolytic virus, a virus that kills cancer. So that's another thing, and I think there's going to be a lot of oncolytic viruses coming down the pike. I mean, there's already a bunch that are in sort of early phase testing, and I think that these things have the potential to make a big difference for some patients. Oh, so I was talking about TLR before, but there's actually an FDA approved TLR agonist. So remember I was talking about William Coley and the bacteria and those receptors called toll-like receptors. There's actually a cream for some skin cancers. It's called a miquimod. It's a TLR7 agonist, and it really works for certain skin cancers. So you know, maybe this sort of thing will play a role in sarcoma eventually. Oh, and lastly, I think, you know, we're kind of getting to the, because I want to make sure you guys have time for questions, but the BCG is a very old treatment. It's FDA approved forever for bladder cancer. If you have bladder cancer that is invading into the muscle, your urologist could, one thing they could do is go up there and inject this bacteria, killed bacteria product into the cancer, and a lot of the times it will make a big difference in helping to eliminate the cancer there. That's called BCG, BCG treatment. So that's an established standard of care for certain types of bladder cancer that is an immunotherapy. So I want to say thank you very much. I want to make sure you guys have time for questions. I just kind of want to, because there's no summary slide here, I just kind of want to review a couple of things that we talked about. So T cells are the main drivers of immunotherapy, but in leiomyosarcoma, there's a number of important cells, and macrophages are one of the critical cells. There's a number of different important types of immunotherapy that we talked about. We talked about PD-1 inhibitors. That's kind of the backbone of immunotherapy in the current era. And we talked about CTLA-4, which is another checkpoint inhibitor that can sometimes be used to bolster up the PD-1 effect. We talked about cellular therapies, gene-engineered therapies, especially CAR T-cell therapies. These don't play a role in leiomyosarcoma right now, and I'm not aware of any trials that are happening this moment, but I think those trials will be coming over the coming years. We talked about vaccine treatments, and I like the cellular therapies. I think these the new generation of cancer vaccines are going to be happening over the next decade. It's going to be really exciting. We talked about TLR agonists. We talked about TVEC, an oncolytic virus. So we talked about all sorts of different immunotherapies. I think this was kind of like a basic overview, but I hope you learned something, and I'm happy to take any questions.
Hey, if you have questions, you can unmute yourself and ask them, or you can write them in the chat box, but I'll start us off. So Dr. Pollack, thank you. That was really a lot of information.
It was a lot. I'm sorry if it went a little quick, but you know, I'm here for questions. So
yeah, so we'll break some of it down. Well, a lot of us got very excited back in 2006 with the discovery of a lot of macrophages with Matt Van der Rijn in the LMS tumors. And we got excited about anti-CD47 trials, and they still are out there. We haven't gotten leiomyosarcoma into any of those that I know of, but have you heard the latest on how those are doing and for which cancers? And
yeah, so there, you know, the problem with anti-CD47 was So the antibodies for anti-CD47, they worked really good at hitting the target, but they had to really be injected into the tumor. And there was some toxicity in terms of especially people's platelets. And I am aware that there are some very exciting new anti-CD47 products that are going to be, that are already kind of in clinical trials, that have different mechanisms that allow them to be infused intravenously, and you know, different kind of like technologies that sort of take, you know, it won't be an anti-CD47 until it gets into the tumor, and then you know, a piece drops off and it becomes anti-CD47, right? So I think those technologies, those new products really do have the potential to be groundbreaking. Yeah, I do think it's a shame that leiomyosarcoma isn't kind of at the forefront of these things getting pushed forward, but I am hopeful that some of these products, you know, once they see some activity, that leiomyosarcoma could be. I know some of the companies know about leiomyosarcoma, so I've talked to them about it, and I know that they know leiomyosarcoma is a good place to look for a response.
Great. Just so the rest of you know, Dr. Matt Vanderine at Stanford, he actually did a CD47 trial with mice and leiomyosarcoma and got responses. So we've been waiting our turn for a trial patiently, or maybe I should buy stock. I'm not so sure. Any other questions? I think Julie has her hand up. Linda has her hand up. Yeah. Julie, has your hand up?
Yeah, my hand is up. Yeah, I just wanted to ask, I know you were, yeah, just in general, if there's anywhere in the world that's doing this routinely already with these cancer vaccines, like outside of the clinical trial setting. I know there's very few trials right now for those people with LMS, but is there anywhere that people can go to try these kind of cancer vaccines, or the other TVEC that you mentioned?
Oh, well, TVEC, there is a trial going on right now, and I know that it's at a number of sites. The lead investigator is at Mayo Clinic. He's Stephen Robinson. And I don't know the details about it on the top of my head, but that is a trial for sarcoma that I think is enrolling now for TVEC. For cancer vaccines, I don't know of a personalized cancer vaccine trial open right now in sarcoma. There are some trials like that in melanoma and some other cancers, but they haven't gotten to sarcoma yet.
Okay, thank you for that answer. It would that TVEC trial be accessible for Canadians? Do you know?
Yeah, it's always tough with that. That's a little bit of it. So that then you're talking about really an insurance issue.
Right.
So, you know, it comes down to, I don't know. So when I was in Seattle, a lot of the times we'd have patients in Vancouver and they would want to participate in clinical trials. And I would always try and write my trial in a way where patients would be required to do as little of standard stuff in the US as possible, because the standard stuff usually you get a bill for, right? So that's the way the trial budgeting works, is we have to say, what are we going to bill the patients or their insurance companies for? What's the trial going to pay for? So we usually say, like, standard things like your routine labs, your insurance company should pay for, and other stuff the trial should pay for, like if it's specific for the TVEC. So I always tried to make it that things like CT scans, routine labs could be done wherever. And I think if it's like that, then probably you'll only, you know, there will be it won't be too expensive to get the treatments someplace like the Mayo Clinic, but you know, that you'd have to kind of get into the weeds about it.
Okay, thank you.
Linda?
Yeah, I'm actually Linda's son-in-law.
Yeah, you're not Linda, are you?
I'm sitting in for her. She had to step away. I'm her son-in-law. I'm the one who actually has leiomyosarcoma
Okay.
for 5 years. I'm on trabectedin. I was just curious, is trabectedin killing all macrophages or just the anti-tumor ones or the pro-tumor ones, since there are 2 types of macrophages in the tumor microenvironment? And my other question was just, what's the target for the CAR T cells that they're doing at Stanford in osteosarcoma? I was just curious what the target was.
B7H3. B7H3.
I'm sorry, B7.
H3. B7H is in Harry 3.
Is that something specific to only sarcoma? Like healthy cells don't have that?
Yeah, so all kinds of tumors can have it. Healthy cell, I mean, it is a self protein, so it's a protein that has the capacity to be on healthy cells. So there is, you know, I think people have been a little bit slower to move on it than they have some of the other targets. Like, so for example, the CD19 CAR, I think people were pretty confident CD19 was going to be very restricted to those B cells. So they said, well, even if we kill all the B cells, we could do a bone marrow transplant or something like that, right? B7H3 is, it is specific for cancer. People have seen it in some autoimmune situations. For example, myositis is a problem where people get autoimmune muscle inflammation, and it's been observed. It's probably, we think its function is sort of like PD-L1. So it's something that it functions as an immune checkpoint, but it's not something that's commonly seen. So yeah, I think there was concern because there was one sample that somebody saw one time with from an asthmatic where there was some B7H3 in there, right? And that's a common problem. But I think tons of asthma samples have been looked at now, and they haven't seen B7H3. So I think that was like it's not common. I think that it's very cancer specific. And now there's been some trials targeting B7H3 with an antibody treatment. So there's a company called Macrophages. They make an antibody against B7H3. They've treated a lot of patients. They've even had some sarcoma patients on their trial, and it's been safe. And they have seen some responses with just the antibody treatment. But I think that, you know, putting that antibody or putting an antibody onto a CAR is going to be a whole lot more effective.
Yeah, because of all this, that sounds very effective. I just have one other question. The trabectedin, is that, you mentioned that might be killing macrophages in the tumor microenvironment. Is that killing all M1 and M2 or just the anti-tumoral ones?
It's definitely more effective at killing M2, but I think it probably kills some M1 too. Yeah.
Okay. Yeah. Great. Thank you. Thank you very much.
Yeah, sure.
It sounds like it would be a big side effect if it kills the good ones.
Well, I mean, it's immunosuppressive, like your whole white blood cell count like drops on trabectedin, right? So I would kind of, the way I sort of view this macrophage effect is just like trabectedin affects all sorts of lineages. And macrophages, they're kind of white blood cell, right? But, you know, and trabectedin affects your platelets. Some people, it's like a big problem, their platelets, right? Some people, their neutrophils is a big problem. But, you know, these macrophages, these immunosuppressive macrophages are kind of like, you know, fortunately, they're the white blood cell that gets hit the hardest. You know, the good macrophages get hit too, but not quite as hard.
The questions?
I have one more.
Okay.
Or I have one. So my son, who's 28, has metastatic vascular LMS, and I know a lot of the studies are, you know, uterine. But his oncologist is thinking kind of outside of the box, and he's taking Pembro and Olaparib because the tumor has a high tumor mutational burden. Is that something that is going to be looked at, I would say, like more often for people, like that's going to become standard of care? And then my other question kind of ties into that is, as immunotherapy is combined with other things, do you guys plan on looking at like where diet or activity or things that actually are immune system boosters can help a patient with that? Is there anything happening with that?
Yeah, so For sure. So tumor mutational burden is definitely one of these things where when we look at all kinds of patients, we look at lung cancer, we look at melanoma, we look at any sort of cancer, it seems to be a good marker for patient response. We don't really know if it's a marker in LMS, right? But where we think that it probably it is, right? I think it is. I think it will be. So I think if it has a high tumor mutation burden, that sounds promising. I mean, he may. We don't see a lot of. So the other thing that's kind of like a really, and this one is a biomarker, no matter what kind of cancer you have, is called microsatellite instability. I don't know if they looked for that in his tumor.
I'm sorry, you cut out for a minute. What was it?
Microsatellite instability.
I'd have to look at his.
Yeah. So sometimes people with a really high tumor, so because leiomyosarcomas tend to not have that high a mutational burden. I mean, they can high for LMS is sort of mediocre for everything else, but microsatellite instability is something that any tumor could have. It could be like the root cause of the tumor, like on a genetic, from a genetic point of view. If he has that, then those patients are very likely to respond. And if he has the high TMB, I would say most people, it's like only 3% of sarcomas have it, but yeah, it is. I think probably younger patients are kind of more likely having it, and he's young. He's got a high tumor mutation burden. That might be. I wouldn't be surprised if he had that. The olaparib, that is something that's exciting. I mean, that's what I was talking about with Dr. Mova. She's looking at that, right? The PARP inhibitor. I think it is something that's exciting. That is outside the box. That's something that we're kind of. I definitely am recommending people go on clinical trials to try that combination if there's a clinical trial near them. So I think that it's promising. The vascular leiomyosarcoma, though, I mean, only a third of leiomyosarcoma is uterine, right? I don't know if he had a retroperitoneal tumor or from his extremities, or
it was extremities and radiation induced, and we have a TP53 germline and a CHEK2 germline, so we're Li-Fermini patients. So there's lots of genetic
going on there, but radiation induced sarcomas, we're also seeing a lot of nice responses with checkpoint blockade. So I think those are more likely to respond.
Great.
Yeah.
Lauren, do you want to ask your question? Because
sure,
I
yeah,
can't read that big word there.
I know. I'm sorry. Well, I typed them out to you, but I can make them shorter here. Okay. One of my questions is, my questions are really material questions, is I've had the experience, I was put on an immunotherapy trial, and it kept growth away everywhere, except at the same time, I had a metastasis to my spine and now kind of in my bones. Okay. So what I was wondering is, is it usual for chemos to be, even if it's the same tumor, but that the location of the tumor actually makes them more and less vulnerable? So that was my first question. And my second question was, how do you think about the switching of different kinds of chemotherapy? Like I'm on trabectedin right now. I mean, I just had my first infusion of it. And then like at some point, my doctors kind of say, well, now I'm going with my gut and I think you should go on an immune therapy thing here. I was on Votrient, and then something grew in my spine, but not anywhere else, so I'm just kind of curious about whether switching different kinds of treatment has something like a cocktail effect.
Yeah. So my philosophy on switching treatments is that, look, we've got a lot of options in our pocket. We can, I'm usually able to kind of come up with something new, but if you're, there's so many really good therapies, and then, at the end, there are therapies that are kind of sometimes they still work for patients. I've seen some patients on treatments that aren't as good and that the world would kind of consider not so great, but they still respond great. So, I'm always optimistic, but still, whatever treatment you're on it because your doctor kind of thought that, hey, look, this is a treatment that, has a good shot. So my philosophy is, let's try and milk as much as we can out of each treatment that you're on. So, even if, things are basically stable, but maybe they're growing just a tiny bit in the wrong direction, well, let's just watch it closely. And it's not really progressing, but it's, starting to move in the wrong direction. We could just stay on top of it. What if you just have one tumor that's growing in that situation? Well, maybe we could radiate that tumor, right? Maybe you can still kind of continue benefiting from that drug. Because, if you get, if we give you an extra 3 months on one drug and an extra 3 months on another drug, extra 3 months on another drug, before you know it, you had an extra year. So that's how that's kind of my approach to it. So I don't rush to get people off of these things. I think that we try and kind of we stay we don't want to fool ourselves with anything, but if things aren't basically under control, then they're under control. In terms of different locations and things like that, well, I mean, there are some locations that have, it's kind of like yes and no. So well, for sure, the brain and spinal cord are very different than any other location. In the bones, it is also there. Those lesions also, I think, are a little different. I usually put those patients on. If you have a lot of bony disease, I usually put people on a bisphosphonate to help strengthen their bones and prevent fractures. And I think there is probably, this is kind of like a little hand wavy, but there, like back in the day, there was a time when people thought these drugs that would strengthen your bones might also slow the progression of disease in the bones. And I kind of think there's something to it. That's not proven. That's just kind of like my personal take on things, and what we've seen kind of from some of the science, and I think there's probably something to it. Beyond, so there are some specific things like that, but then there is kind of your particular disease, and you may have a tumor that is behaving differently from your other tumors. It's just a different tumor. It's mutated, something's different about it. And, there are definitely a lot of patients out there who their disease is all responding to the treatment, except for this one particular tumor that is behaving differently. And if that's the case, I like to, if I can, radiate that tumor or have it removed, or, do something to treat that tumor. That's my general philosophy, because again, I don't want to give up on that drug that is working for all of your other disease.
Sounds good. All right, Lauren? Yep, thumbs up. Okay, so we have a question in the chat asking if you've heard of Dr. Ralph Kleef and his work on hyperthermia, and your opinion on that.
So I think I know what you're talking about. I mean, hyperthermia is something that is, long been interesting, and it's used in a variety of different ways. For sure, there is well, there are different situations where hyperthermia is being used in very specific ways. So, for example, people are using hyperthermia along with intraperitoneal chemotherapy, or with therapy for an extremity tumor, where they're able to kind of basically separate off. I mean, it's a little bit grotesque the way, to get into the details, but separate off the blood supply from an extremity from the rest of your body, so that they can give like a much higher dose of chemotherapy and a much and hyperthermia as well. So like hot, hotter than your body would normally be able to handle, and that's separated off from your body. And I think there is,
Turn that off, honey.
I think there's a lot of reason to think those sorts of approaches have benefits, and I don't think it's something that is. I think it's something worth studying. I haven't kind of seen anything that has kind of given me conclusive evidence that this is the right direction to go. But you hear anecdotes of people that have been treated with these sorts of strategies that did better than you would have expected. So I think that hyperthermia has some patients out there like that. So I think that there probably are some patients that this sort of approach helps. Yeah.
Okay, thank you. Kim, do you want to answer your question or do you want me to do it? Where are you?
I can ask real quick.
Okay.
So my mom has metastatic LMS that's spread from the pancreas to the liver. So she's on systemic treatment now. That's with Gemtex and a checkpoint inhibitor. So she'll receive the Gemtex with immunotherapy for 8 cycles. And then after 2 years, just the after that, she can go on just the checkpoint inhibitor for 2 years at max. And I'm wondering, like, at what scale of the tumor regression can we feel safe to just go with immunotherapy and not with the chemo combined? Because we don't know which one is shrinking the tumor, if it's the chemo or the immunotherapy. And the radiologists, like one of her liver tumors is shrinking while the other one in her pancreas, which is the primary tumor, has grown slightly. But the radiologist thinks it might just be necrotic tissue that's making it appear that way in the scan.
Yeah. So is she on a clinical trial?
Yes, at MSK.
Mm-hmm. But they're giving you the option of when she would come off the Gemtex. Usually a clinical trial is pretty rigid about that sort of thing.
They're not giving us an option. I'm just wondering, like they're going to give her for 8 cycles, so 4 months. And after 4 months, they're going to take out the Gemtex. And I'm kind of just worried that when she's off the Gemtex and just on the immunotherapy alone as a single agent, that, that's risky.
Yeah. So, the way I would think about it is, when I showed you those curves of patients that had been treated with checkpoint inhibitors, and you saw that some of those patients had like really, they went for a really, really long time on the checkpoint inhibitors, right? Mm-hmm. And that's really your goal, right? You want her to have this kind of really durable response. I mean, the best thing about checkpoint inhibitors is, look, there are some patients who have a lot of toxicity from checkpoint inhibitors, right? There are, but most people on checkpoint inhibitors, it's very well tolerated. So I always tell my patients, there's probably about 50% chance that you won't have any significant toxicity at all. And then if you do have toxicity, it's more likely than not, it's going to be mild stuff like rash that you can treat with creams or just mild diarrhea or something like that. And it's really just a small fraction of people that have significant toxicity. So imagine if she was a responder and she was able to go 2 years with just like that mild toxicity and not having chemotherapy, where Gemtex chemotherapy is really tough. You've got to be going in 2 out of every 3 weeks, and it's no joke. So, I mean, that's the point of the trial, right? Is to get her to that place where she's not on the Gemtex, where she's having hopefully a nice response to just the checkpoint inhibitor, right? And the reason why they're giving her 8 cycles is, one, they think maybe there's synergy between the pembrolizumab and the chemotherapies on Gemtex, and there are some reasons to think those chemotherapies might help. But there's another thing too, which is just that the checkpoint inhibitors take a while to kick in, and the Gemtex. So maybe it's both helping to stimulate the checkpoint inhibitor, but maybe it's also helping her get over this bridge where you're waiting for the checkpoint inhibitor to kick in. So I would say that if she's got a shot at just being on a checkpoint inhibitor, go for it. I think that sounds like a good idea. And if she progresses once you take off the Gemtex, well, then you learn that, Gemtex is probably, I don't advise people to go on and off chemos willy-nilly. And, we were just talking about that a second ago. But, this is a case where I would say make an exception. If she progresses when you pull off the Gemtex, just go back on it. That's what I would say.
Okay, I was just curious if it was too toxic, the combination of chemo plus immuno, because she also went into the hospital for sepsis, and I don't know what caused that, if it was the immuno or the Gemtex. I don't.
Probably the Gemtex, because the immunotherapy, it's not sepsis isn't like it's not kind of one of the main things that it causes. People can have it, but usually it's the Gemtex that's making you immunosuppressed, right? The immunotherapy doesn't really make you immunosuppressed, right? It revs up your immune system. Sometimes that can surreptitiously lead to an infection, but that's not a normal thing. That's like a really weird, unusual thing. The normal thing though is I talk to you a lot about T cells, right? But the cells that are most involved in fighting bacteria, the kind of bacteria that cause sepsis, are your neutrophils. That's the ANC. Those are the cells that you're pumping up with Neupogen and Neulasta. Those are the cells that you really need to fight against sepsis. And the checkpoint inhibitor isn't depleting those. The Gemtex is depleting those.
Okay, thank you.
Good to know.
I have a question.
Go for it, Nancy.
Okay, why would a history of radiation pneumonitis exclude a patient from going on a trial with checkpoint inhibitors?
Yeah, so I mean, pneumonitis, right? Okay. So, all right, this is what the people that wrote the study are thinking. It's not an endorsement of it, okay?
Okay.
Pneumonitis means that you had inflammation in the lungs, and it could be that whatever, you had an inflammation in your lungs, now it's settled down, but maybe there's something subclinical that's smoldering there, and if you give a checkpoint inhibitor, it's going to flare back up. That's what they're worried about, because inflammation in the lungs is one of these things that you don't want to play around with, right? If you're writing a study, you get one patient that has really bad inflammation in the lungs, they have to go to the ICU, right, to be on a breathing machine while you give them steroids to get off it. That, like, from the peoples that are designing the trials, they're thinking, what are the worst things we want to avoid? That's going to screw up our whole study. That's how they're thinking about it, right? Now, from your perspective, where you're sitting, it's probably pretty unlikely to happen to you, right? Especially if your case of radiation pneumonitis wasn't that bad, and you're totally over it now. Maybe it's been like a couple of years since you had it. So I don't want to say that it's right, but if you're just trying to kind of understand why they put it that way, that's why they put it that way.
Okay, so that doesn't necessarily mean I won't ever be eligible to take checkpoint inhibitors.
No, for sure. There are studies out there where radiation pneumonitis, or a distant history of radiation pneumonitis, is not a contraindication. So, it may be that the specific study that you're looking at right now has a contraindication, but I wouldn't say that's kind of like a universal thing, especially if it's, if there's some time that's passed since you had your radiation pneumonitis.
It was probably less than a year, but I was really hoping to get in with Dr. Wilkie at UC Health, but unfortunately, due to that, I was not able to.
Well, the good news is you may down the road be able to take it. So
Thank you.
Yep.
who knows? I have a question. What are you working on now, Dr. Pollack? Do you have any trials in the mill?
Yeah, I mean, I have, look, it's so, I've been in Chicago for about a month, and it's everything is kind of booting up from scratch. So, I mean, Mark Goldnick he's great doc, but I've got a lot on my plate. Like, there's so many patients in Chicago that we got to see, and we've got to get, I've got trials that I want to do, but I think kind of the first step is we got to get all the kind of like, there's like national trials that are going on right now. We want to get those up and running. There's not really anything. I mean, we have Immunotherapy for LMS, right? But there's not that many trials right now for LMS. But for other sarcoma subtypes, we're trying to get that going. There are some trials that are kind of like, I think are sort of baked and ready to go, and we kind of are just sort of trying to put some of the final pieces in terms of not getting them open, but in terms of until we can like actually start to think about, well, what would we need to do to move forward. So they're really kind of in the starting point, right? We're really trying to just move forward, but there are some things that we want to get moving. But there's a lot going on. Like I came in, like I have a huge lab. It's empty. I don't think I've ever had been in a totally empty lab like that for any, like I had to, because my lab in Seattle, it was sort of like it was the same lab I trained in. I just kind of grew up there, and I just stayed there, and eventually they put a sign on the door that said it was mine. But it was, this is kind of like I come in, I've got to buy all the kind of basic stuff and get things going. So it's a lot, I'm like trying to hire people. So there's a lot it's gonna take a year, I think, really before we're kind of like firing on all cylinders here, but we'll get there. And we're definitely gonna plan some really amazing trials for LMS when we do.
Yeah, we'll fill them. I've been sending people your way, actually, in Chicago.
I appreciate that.
Yeah. And while we're at it, what trials do you see as that are out there right now that are promising?
Yeah, well, so Nancy just mentioned the Bree Wilkie study at University of Colorado, the doxorubicin ipi-nivo study. I really like that trial. I really like the olaparib study with the Dr. Mova study at Memorial Sloan Kettering. I didn't realize that Memorial Sloan Kettering had that GemTex pembro study, but I have been kind of involved in a similar study at with Robin Jones. I helped him design a study in London that I've been really excited about and kind of following from afar. So I like that concept of GemTex and checkpoint inhibitor. So I think that also sounds like a good study. And then, as I was saying before, I think there's a lot of really exciting stuff coming down the pike. I think there's going to be a bunch of trials, like for example, the trial at the Mayo Clinic we were talking about before. It's a multi-center trial led by the Mayo Clinic, where people are using TVAC in combination with the checkpoint inhibitor. And I think there's going to be a number of other oncolytic virus therapies that are going to be available for sarcoma patients. So I think there's a lot that's happening.
Great. Charles, I saw you raise your hand. Do you want to ask something? You're muted.
All right, this is gonna be the last question because I do have 5:30.
Okay,
Yeah.
Last question, Charles.
Dr. Pollack, this is Charles from Anchorage, Alaska.
Oh, hey.
Yeah, you probably remember me from SCCA.
Yeah.
Since I went palliative care, I started looking for things that would really maybe help. And right now I'm on Right to Try with leronlimab, which is by Citadine. They have a basket sarcoma trial down in San Francisco, I think. But I just wondered if you were aware of that Ccr5 marker and anything else that was being done with that.
Well, Ccr5 is a really important marker for certain types of CD4 cells. And I think it's something that, it stimulates exactly the kinds of CD4 cells that you want to be fighting cancer. But I don't know much about that drug, to be honest. So I'll be curious to hear how you do. If you do well, please let me know, and I'll try.
I'm still in touch with.
I'll see what I can do to try and push that drug forward and get some other sarcoma patients on it.
Yeah, I'm still in touch with Dr. Cranmer. He recommended I go trabectedin, and up here I couldn't get a pump to take home, so we looked at that and said it's easier to do this, the leronlimab, since it's just a shot like Neulasta. And it's a large molecule, so it goes in pretty slow rather than an easy shot like Neulasta. But it seems to be working great so far. I mean, we haven't had the first CAT scan, which comes up in about 3 weeks, but I feel great and has zip side effects.
Hey,
So-
we like that.
Well, I'll be anxious to hear how you do. Maybe next time I talk to Lee, I'll ask him how you're doing.
Okay.
Best of luck to you on
Congratulations
that,
on your,
Charles.
on your job.
Thank you.
Yeah.
Good. All right.
So
Well,
I want to thank you very
sure
much.
thing.
It was a lot of information and we really appreciate your time for this.
it's my pleasure. Really glad to be here. Okay. Thank you, everyone.
All right, thank you.
All right. Okay. Bye.